If your breed club is interested in working with us, do reach out at breeders@embarkvet.com, and we really always enjoy doing things like this. So first, a little bit about me. My name is Casey Erlich, and I've been with Embark for about 4 years now. I studied biology undergraduate and then went on to study biomedical forensic science in graduate school. This means that my prior work in grad school was actually in human forensics and DNA analysis, but I ended up migrating over to dogs afterwards, and I think the appeal there is probably obvious. I'm a research associate at Embark, and my specialty is the genetics of phenotypic traits such as coat color. Our mission at Embark is to improve the life and longevity of all dogs through science and technology. We know that this often starts with breeders, so we offer tools and services to help the breeder community manage the genetic health, traits, and diversity of their breeds. So, given that the Sloughi is a very interesting landrace breed, there might actually be a particular interest in Embark's story and how the company was founded. Our co-founders, Ryan and Adam Boyko, conducted field research in over 40 countries, and they sampled free-roaming, free-breeding dogs, which we call village dogs. There's a connection here because breeds like the Sloughi are derived from these village dog populations, and these dogs can actually tell us a lot about the evolutionary history of dogs, so they're of particular interest to study. As a company, we are actively innovating, and we're researching the genetics of canine health. We are also a research partner of the Cornell University College of Veterinary Medicine. So many people want to know, why are we different? What sets us apart from other canine genetic testing companies? Well, one key point is that we actively seek to partner with breed clubs and organizations, as well as researchers and other professionals in the field. As for our test itself, we use a high-resolution research-grade platform that queries over 200,000 genetic markers. What does this mean? Well, we're looking at over 200,000 different locations across the genome, and many of those locations are associated with health conditions, traits, or other areas of interest. And when taken as a whole, this information also gives us unrivaled accuracy in determining genetic diversity and breed ancestry. So we will go a little bit into the nitty-gritty of how that works shortly. But historically, many breeders have utilized one-off tests that will query a specific mutation of interest. And our approach is different. So by gathering all of these data at 200,000 locations, we're able to drive discovery and research. And you can too. So what we do is we actually provide every user with their dog's raw genetic data so that it can be shared with other researchers, added to databases, and more. And the next question is usually what's included in each test. So as a quick overview, each test includes genetic health variant testing for over 200 mutations, coat color and trait results, genetic coefficient of inbreeding, which we will talk about more later, optional participation in research surveys, and as I mentioned, as well as your dog's raw data for any other research you want to participate in, genetic counseling, and event sponsorship opportunities. And I also want to mention that I know many here today are joining us from overseas, so I wanted to share that we do ship to over 240 countries, and this includes Morocco, Tunisia, and Algeria, which, as you know, are 3 of the Sloughi's countries of origin. So though we have limited time here today, so I can't go through everything, I included some great resources here on subjects of interest, and we have a lot of information on our website that's absolutely worth exploring if you enjoy what you hear today or are just curious about genetic testing. So first, I'm going to walk us through some information on testing technology. And I know there's likely varying levels of familiarity with genetic testing here today, and it's an interesting topic. So certainly if you have any kind of follow-up questions about this information, definitely share them in the chat and we will loop back and make sure they're answered. So remember a few slides back when I mentioned that we're querying or testing over 200,000 different locations? Well, each of those locations is referred to as a single nucleotide polymorphism. And that's a long way of saying what we abbreviate as SNP or SNP. And it just rolls off the tongue a little better. And many of you probably know or remember that DNA consists of 4 different base pairs or nucleotides. And we usually refer to these as A, T, C, and G. And the really helpful thing about these base pairs is that A will always bind to T and C will always bind to G. And if you remember, DNA is double-stranded, right? So the opportunity for this, this kind of predictable binding is there. And the reason I mentioned this is the SNPs that I mentioned above actually only consist of a single base pair. So one SNP might be a T, one SNP might be a G, and so on. So our testing is done on a piece of technology called a SNP chip. And you might also see or hear it referred to as a microarray. It's the same thing. And this is actually a little wafer-sized chip pictured here. And it's full of what we call probes. So they're called probes because they are probing for specific information in a very literal way. If you remember what I just mentioned about how base pairs bind to each other in this kind of predictable manner, we can actually use this to our advantage. So each of the probes on the chip contain a short sequence of DNA. And we use these short sequences to kind of fish for the complementary sequence found in your dog's DNA. And the end result of this is that we're able to determine a dog's genetic status at each of these many locations. So we essentially end up with this readout of a dog's genotype information. And as I talked about earlier, that can be hugely beneficial to research, especially with a large sample size, because it allows us and others to discover associations between a dog's results at any of those locations and a dog's phenotype or physical appearance, or other things that are being expressed, like health conditions, which can include appearance. And many discoveries have actually already been made using this technology, both in dogs and in humans. So I mentioned that when taken as a whole, the sort of constellation of these SNP results can give us a very accurate picture of what a dog's genetic ancestry looks like. So as a result, many wonder if Embark or other genetic testing companies are able to authenticate whether a dog is purebred. And the answer to this can actually be a little bit complicated. The short answer is no. No breed ancestry test can certify that a dog is purebred. But the reason for this is because there's really no scientific designation of what a purebred is. And this is because that purebred designation does require registration status or pedigree records most of the time. And these are things that cannot be picked up on a genetic test. So while we do encourage owners and breeders to use DNA testing to learn more about their dog's ancestry, as well as health traits and relatives, owners looking to register their dogs with various organizations will need to submit their pedigrees to the appropriate registry body for that certification. Worth noting, these registries typically do not include the requirement that the dog be certified as a single breed from a DNA testing company. And as you might imagine, ancestry testing for a breed like the Sloughi can also be a little bit more complicated. And I will talk about this more in depth later, but landrace breeds can be harder to identify because of their vast genetic ancestry. So it's a lot easier for us to kind of figure out the genetic signature for breeds that have a smaller population and more what we call homozygosity. So they're more genetically uniform. And I know there's a lot of interest in physical traits. So what I will do is I'll walk through some of them here. And I also pulled some information from the Sloughies we've tested so far, so we can talk about what coat color genotypes we've seen in the breed. So first, here is an example of what a Sloughi's coat color results might look like. There is certainly variation, so it's definitely possible for Sloughi's to have results that are different from this. And we also test for some other patterns and traits that are not captured in the screenshot. I'm just going to wait a beat so everyone can kind of take a look at what's here, and then I'll move on to the next slide since there is a better visual of how all of these genes interact with each other, and they do interact with each other. All right, so don't be too intimidated by this. But the coat color genes that are likely of most interest for Sloughi's include the Elocus. So I'll talk about that first. This will determine whether a dog will have what's called a melanistic mask, right? You can see it on this little dog illustration here. And it's really just a dark muzzle. So this gene also determines whether a dog will be solid red or whether other coat color genes will be expressed instead. So the way to think about all of these kind of combinations of letters is that every dog is going to be inheriting half of their DNA from their sire and half from their dam. And so you'll see kind of combinations of two different letters or pairs of letters. So in this case, we have this dog that inherited an EM from either their sire or their dam, as well as a small e, right, from their other parent. And in this case, if a dog has one or two EM alleles or variants, we would expect them to be expressing this, this dark muzzle or melanistic mask. So the other allele that's of note on the Elocus here is this capital E. And if a dog has typically one or two of these kind of uppercase E's, that essentially means that you can ignore the Elocus results and you want to next look at the Klocus. So what the Klocus does is it determines whether a dog will be solid black, brindle, or whether the Elocus will be allowed to be expressed. So you might notice there's kind of a dominance hierarchy here between these different loci. So the sometimes confusing thing about the Klocus is that there actually is not a test for brindle yet. So as you can see here, dogs can have 3 different results at this locus. So there's this result of KY, which you could probably see it feeds into the Elocus, right? So if a dog has a KY result, you would then look at the Elocus to figure out what other pattern might be being expressed, and I'll get there in a moment. And then the other 2 results here are KB, which results in a solid black dog, right? And if a dog actually has this allele for a mask, but they're solid black, then you don't actually see the mask. But that mask can then be conferred to any offspring, and it might show up later. And then there is also this result of KBKY, which is the interesting one here. So This can both mean that a dog is solid black, but it could also actually mean the dog is brindle. And the reason for this is we actually see that, despite there not being a definitive test for brindle, we do see that all brindle dogs that have been tested with us do have this KB-KY result. So in other words, KB-KB will be black, KY-KY will not be solid black, nor will it be brindle. And you want to look at the Elocus, and then KB-KY could either be black or brindle in the Sloughi. So I've mentioned the Elocus a couple of times, and this locus will determine some types of patterning. So you might be familiar with tan points or sable, and sable is kind of the genetic term for when a dog has a tan coat with some darker hairs that are dispersed throughout it. And I'll also point out that each breed has their own breed-specific terminology for these coat colors. So I'm using the terms that we usually use when we talk about it in, kind of the field of genetics, but certainly each breed has their own language surrounding these colors. And what's kind of interesting is that all of the Sloughies that we have tested so far are either sable or black and tan at this locus. Now, it's also worth mentioning, and you of course know that not all Sloughies are black and tan or sable, right? They're not all expressing it. And that's because these colors or these patterns won't be expressed in a solid black, solid red, solid brown, or brindle dog. And then also on top of these three loci that I mentioned and talked about, there are two others that play more of a modifier role, and these are both relevant to the Sloughi. So there is the B locus, which will convert all black pigment to brown. So it actually acts on top of these other results. And the D locus, which is kind of similar in that it is a modifier, and it lightens or dilutes pigment. So most Sloughi do not have these mutations, right? So if a dog has two capital Bs here, they are not carrying for brown, nor are they expressing it. If a dog has one small B, they are carrying for brown. And if a dog has two small Bs, they would be expected to be expressing brown. And then dilute, again, is very similar. So if a dog has two uppercase Ds, they are not carrying, nor are they expressing dilute. If a dog has one capital D, one lowercase d, they're carrying for dilute. And if a dog has two lowercase d's, they would be expected to be expressing dilute. And in some breeds, you actually very rarely have both being expressed at once. And I believe the only breed this is a common thing in is the Weimaraner. So it's kind of that very light brownish color known as Isabella. And what might interest you guys is that, as I said, most Sloughies don't have this either the brown or the dilute mutations, but we have actually tested a couple Sloughies that have one copy of the dilute mutation, as well as a couple that have two copies and therefore might actually be expressing dilute. And then similarly, we have tested a couple of Sloughies that have one copy of the B locus mutation for brown, so they're carrying it, but we have not yet tested any who have two copies yet. And of course, given that there are carriers for brown, it is likely there are dogs with the brown mutation that are expressing it that just exist outside of our sample size. And the other thing worth noting is that it's actually quite common for these mutations to exist in a population, aka a breed, even when they're not accepted coat colors. And this is because, especially in the case of brown and dilute, they're actually recessive. So it requires 2 copies to be expressed, right? If you remember these 2 lowercase b's and 2 lowercase d's. So they can effectively hide. And then as well, Sloughies as a landrace breed will typically have a large and diverse founding population. So there's more opportunities for these rarer or more uncommon mutations to be in that gene pool. All right, so I want to quickly mention some savings on Embark kits before we jump into the Q&A. Our Embark for Breeders kits usually retail at $179, but we do have some special reduced pricing available. So we offer litter packages, which are savings when you're testing a litter, as well as breeding packages when you're testing both a potential sire and dam. And then most excitingly, Sloughi breeders have a special club offer. So if you order through your club portal, you can purchase Embark for Breeders at $129 per kit. And then, of course, for the latest on sales and product updates and more, follow us on Facebook. We really post a lot of great content on there that you won't want to miss. And it is time for what many of you have been waiting for. We received excellent questions, and the Sloughi really is such a unique breed, so I'm excited to answer these. And I do want to say I'm sorry I won't get to everyone here, but my team will also be answering questions in the comment section after the talk. And I'm just going to take a drink, one moment. So first we have the question, when will the Sloughi be included in Embark's featured breeds? So in other words, when will we be able to decisively determine whether a dog is a Sloughi or has recent Sloughi ancestry? And as much as we like to, this isn't something that we can give a timeline for. So I mentioned earlier that Sloughi are a very special breed as they are derived from a landrace or village dog population. And this again means that they are especially genetically diverse. And because of this, we do want to ensure that our reference panel is representative of the genetic diversity found within the breed. And this can take a while, but the more Sloughies we test, the easier it becomes to kind of hone in on their genetic signature. If you are interested in helping us learn more about the Sloughi's genetic signature, please do reach out to us. You can email us at breeders@embarkvet.com and let us know you have a purebred Sloughi. And it helps if you include your Sloughi's pedigree and any kennel club papers that you have. And of course, this information will not be shared outside of Embark. So someone asked, what are some measurements that can be made with genetic data? And this is such a great question. I actually broke it down into 2 slides. So there are a lot of different measurements that can be made using genetic data, but I think it's worth highlighting 3 that are most important to breeders. So first, there's what we call COR, or coefficient of relatedness. And this is actually calculated using 2 individuals, and it's the measure of how related the 2 dogs are to each other. So this is done by looking at the fraction of the genome both dogs share with each other. And just as an example, if 2 dogs share 50% of their DNA with each other, the implication is that the 2 dogs are likely either parent offspring or full siblings, right? So just like in humans, you're going to share 50% of your DNA with each parent, as well as 50% of your DNA with each sibling, each full sibling. And this can actually get a little more complicated in purebred dog breeds because there's often historical relatedness. So relatedness that is caused by inbreeding or line breeding, sometimes even in the very distant pedigree, or even during the founding of the breed. So it might not even be represented in a pedigree at all. And this can actually cause the COR to be higher than we would statistically expect in other species or in an outbred population, such as with many mixed breed dogs. So, for example, you can have full siblings that have a coefficient of relatedness that is 65% in some breeds, and that's completely normal. Now, there's also genetic COI, or genetic coefficient of inbreeding. And this is a calculation that's done on a single individual instead of a pair. So it's a direct measurement of the amount of a dog's genome that is inbred. And you might be wondering how we can determine this without testing the parents, right? Dogs are going to inherit one set of chromosomes from their sire and one from their dam, as I mentioned before. And if each set has a lot of identical regions in common with each other, so DNA that is both on the set of chromosomes from the dad and the set from the mom, the implication there is that those regions were inherited from a recent common ancestor, which points to inbreeding. So we can actually determine this, and very accurately, by looking at an individual dog's DNA. And then finally, there's kinship, or what's sometimes called expected COI. And this is what many breeders are used to, even now, but especially prior to the advent of being able to genetically test for genetic COI. And this score is calculated on a pair of individuals, like COR, but it's based on the amount or percentage of the genome of identical DNA that we would expect two individuals to transmit to their offspring on average. So this number is based on probability and averages. And this is actually done out on paper most of the time. It doesn't use the genetic data directly. And as a result, it's less reliable than genetic COI because, again, it uses these probabilistic averages of how much ancestry we'd expect a dog to inherit from each relative based on what they actually did inherit. So as an example, in terms of probability, we say that you're probably 25% related to each of your 4 grandparents, right? The same is true of dogs. But the reality of that is that it's possible to be 20% related to a certain grandparent. It all depends on what is kind of being passed down, right? So the kinship doesn't always have the full scope of the situation. How does Embark calculate COI, and what is the value of it? So I touched on some of this, but we calculate COI by looking for these kind of long stretches of identical regions across both sets of a dog's chromosomes. So remember again, one of these sets will be from the sire and one is from the dam. And when there are identical regions across both of them, it can then be assumed that those regions were inherited from a common ancestor, meaning that the parents of the individual share some amount of relatedness with each other. We then calculate a genetic COI based on the length of those identical regions, which we then divide by the length of the genome to generate a percentage. And actually, other factors can lead to apparent runs of homozygosity or these kind of identical stretches. So we have some additional quality control measures in place to minimize the amount of false positives that we see here. And as for what its value is, well, the genetic COI can be a really valuable tool in breed conservation because, as I mentioned earlier, it allows for a more accurate determination of inbreeding than calculations via pedigree. And this is because it accounts for these identical regions that were inherited from further back than most pedigrees. So research has shown that inbreeding can result in decreased lifespan, smaller litter sizes, less frequent heat cycles, and other things that really are not good in the long run. So we are glad to have technology that can help manage inbreeding, and that this technology only continues to improve. Someone asked, is it possible for the long-haired variants to suddenly appear, or is the presence of these genes from some outside genetic source? Oh, there we go. Sorry about that. And this is a good question. So we've had similar questions come up with other breeds before too. So there are a lot of breeds that are typically short-haired where they've noticed these long-haired individuals popping up. And the interesting thing here, so long coats in dogs are caused by a gene called FGF5. And this gene actually has several different mutations that can cause a long coat. The thing is, all of these mutations, at least as far as anyone knows so far, are recessive. So if you remember back when we talked about the B and D locus, when a trait is recessive, it means that a dog will require 2 copies of that mutation in order for it to be expressed. So it's a little harder to get there than a dominant trait. And this means that traits like long coats can effectively hide within a closed population, such as a breed. And this is actually especially true of diverse or landrace breeds, given the larger gene pool and greater likelihood of different phenotypes. That said, it is possible for the mutation to not be among the founders of a breed. So when that's the case, there are kind of 2 possibilities there. The first is that it could be a de novo mutation, which means that it's a new mutation that just kind of arose within the population. That can certainly happen. It's how we got these traits in the first place. And the other possibility is that it could be introduced by outside sources. And we actually haven't seen the most common variant of the long coat mutation in the Sloughies that we've tested so far. But our sample size, as I mentioned before, is not yet representative of the breed's vast diversity. So it could actually be present at a low frequency. Or even at a higher frequency in certain lines or subpopulations. And we do know that there are a number of de novo long coat mutations that have appeared within certain breeds. So, for example, the Afghan hound, which is another landrace sight hound, actually has a different long coat mutation than many other breeds, and that's present at some frequency. So we had a couple of people ask about the genetics of some specific health conditions. So what can you tell us about Addison's disease, irritable bowel disease, symmetrical leukoid onychodystrophy, and lupus? So I have kind of a brief overview of where some of the research sits on each of these. For symmetrical leukoid onychodystrophy, henceforth will be referred to as SLO, 2 candidate regions were linked to this condition in the bearded collie. So, however, these are associations, so there's not yet enough known about a causative mutation to offer a test yet. And the other kind of mystery here is that we also don't know if there might be a different genetic basis for the disease in the Sloughi. So we often see that some genetic conditions are caused by different mutations in different breeds. So they may look the same in a clinical practice. They may be expressed the same way, but they can actually have different genetic causes. And then both SLO and Addison's have an autoimmune etiology that may be connected to a dog's DNA, to a dog's DLA class 2 status. This is kind of the dog equivalent of what's called MHC in humans, but further research is needed on that. And then for IBD, it's likely polygenic with complex inheritance, so impacted by multiple genes as well as the environment and development and other factors. But candidate genes have been identified in the German Shepherd dog. There is more research that has to be done there before a test is developed, or before we know that those genes are causative. And then there has also been a variant for cutaneous lupus that was identified in the German short-haired pointer, but systemic lupus, which is, I would say, what most people are referring to when they talk about lupus, likely also has this complex inheritance that involves many genes. So to summarize, as you can probably tell, there's still a lot left to learn about the genetics of these complex diseases, but we are closely following the research as well as being on the cutting edge of it ourselves. And so this was actually an exciting question to receive. So what can you tell us about wobbler syndrome? And wobbler is actually another disease that there is a lot of interest in, and it's one that we at Embark have specifically chosen to focus on. So we have begun generating data for a study on wobbler syndrome, but we do need your help to continue to collect DNA samples from dogs that have been diagnosed with wobbler syndrome by a veterinarian. And dogs of any breed will be considered here. So all that's needed for this is a cheek swab. There's no cost to participate, and home delivery and return shipping are included. And then there's more information on how to get involved in this on our website. I definitely encourage you to check it out if you have a dog that is diagnosed with wobbler. So this was another good question. So someone asked essentially why they've never seen a Sloughi with bald thigh syndrome, but they have seen Embark tested Sloughi's with either 1 or 2 copies of the mutation that is associated with bald thigh syndrome. And this kind of goes back to what I said about SLO. So where I mentioned that associated mutations may have been discovered in the bearded collie, but we don't know if those same mutations are causative in the Sloughi. So in some cases, dogs that have 2 copies of a recessive genetic variant that is known to increase risk may not express that phenotype. So the penetrance of the variant, which just means the likelihood of the genotype being expressed, may be impacted by factors, including breed. So one thing to note is it's actually even possible that there is a protective modifier in some breeds, or conversely, a modifier that increases risk in other breeds. And in the case of bald thigh syndrome, we know that this mutation is associated with the disease in greyhounds, but whether it's associated with it in Sloughi's is still unclear. So if you have the time, and especially if your Sloughi was identified as a carrier or having 2 copies of this mutation, absolutely fill out our annual health survey or the other surveys on your Embark account, and let us know whether or not your dog is actually expressing this condition. So this is the last question for now. It has been really great to see so much interest in COI. We feel it's very important. And someone asked that, if a dog has a high genetic COI and or low diversity on its MHC or DLA complex, how many generations does it take to ameliorate or improve that? And the really short answer here is that there are immediate benefits to breeders using their dog's genetic COI results to help inform breeding decisions. So, for example, if you have a high COI dog, you can pair it with a dog that will minimize genetic COI in their offspring to avoid additional inbreeding as much as possible. What we do need to consider is longer-term strategies that will reduce the average COI in the breed as a whole, but this will require more research, which is ongoing at Embark, and possibly more coordinated effort across breeders as well. So our research and theory suggests this advice for minimizing the risks of inbreeding. Use the best tools available to avoid unnecessary inbreeding, such as Embark's genetic COI. Test for mutations that are known to be associated with health risk in your breed. Use results as well as your knowledge of your lines to avoid perpetuating a disease in your breed, and keep as many breeding animals of both sexes in the gene pool as possible. Right, thank you so much for joining us today. It really has been a pleasure, and my team will be responding to questions in the comments section, so please do feel free to chime in. And if you have enjoyed this presentation, please give us a follow. So you can join our Facebook group, Embark for Breeders, to learn more about coat color genetics and other topics. Thank you again.